Welcome to everybody who's here in person, and welcome to everyone who is participating remotely and who's watching this after the event. This is our first PLANTPOPNET workshop where we've had data to play with. So we're very excited to be here this week, and I'm going to kick off with a talk that's in the agenda for 15 minutes, but I think it's going to take around half an hour. So let's go. I'm going to give you a bit of the history and my journey to where we've come to, history of the PLANTPOPNET network, and then just a brief overview of where we are. So the aim of PLANTPOPNET is to uncover the drivers of population performance. So it's populations that persist or go extinct in the landscape, and they contribute, this persistence or extinction of populations contributes to a species range distribution, whether ultimately the species persists or goes extinct. Why study within species variation? I mean, this is the heart of what PLANTPOPNET is about. We're studying within species variation in Plantago, lanceolata. We're using it as an ecological model species. It's very interesting. Plantago is very interesting in its own right. It's incredibly common. It persists from the subtropics all the way through to the subarctic under really wide environmental gradients. So it's kind of intriguing how it manages to maintain a level of population performance under very different biotic and abiotic gradients. By studying within species variation, we can shed light on any differences that we see between performance in native versus non-native ranges. And this gives us information on how a species or how a population will perform under new environmental conditions and in new biotic contexts as well. So by using a species that's present in its native range and its non-native range, we can get information on the performance of that species in new environments. This then enables us to develop the tools to predict how populations will respond to new environments in the future. By quantifying the variation in population performance across environmental gradients and how populations actually manage to cope with different environmental conditions, we can develop predictions of how populations will persist or go extinct, how they'll perform at any place along that environmental gradient. And we have multiple dimensions of variation in abiotic space as well as in biotic space. And ultimately, what I'm really interested in doing is predicting population response to global change. So how can we predict which species will persist into the future, under what conditions, and how they'll manage to maintain population performance, or if they're at risk of going extinct, how we might institute management actions to support populations that we want to persist in a particular place, even if their optimal environmental conditions deteriorate. We also need to remember, and this is very important, that population performance is fitness. So understanding survival, reproduction, and growth in the context of the life history will enable us to really think about and develop tools for predicting how plants will adapt to new environmental conditions. Again, this is a huge challenge over the next few decades, predicting how populations will perform under new conditions. So I'm just going to talk a little bit about my personal journey to get to here. Oh, I'm covering the screen, sorry. Just trying to work out where to stand so everybody can see me. Think about there, should be okay. Is that okay for people? Yeah. Oh, sorry, yeah. So I guess my thinking about within species population performance in a global context started out here with this paper, which many people in the room were involved with. So this is a paper published back in 2010. Myself and Satu were the first authors on this one, where we used multiple matrix models for multiple species to look at within species variation and population growth rates. And there was a few things that really came home to me while analyzing these data, and it was that we don't have very much information on variation in population performance through space. So here we've got 50 species, and this is just all 50 species that we analyzed. Each line here is a separate population, and on the x-axis is time. So you can see variation through time and variation in space. And the first thing that should strike you is that those lines are very close together. So there's not much spatial variation going on here from population to population within a species. And in fact, if we look at our most temporally variable species, and these species all show very high negative autocorrelations from year to year, you can see that there's a lot of temporal variation through time, things going up and down, but all of our populations seem to be doing the same thing. And again, this is a problem because we don't have much spatial replication, or our spatial replicates are all really, really close together, which means they're all pretty much doing the same thing. So this was one of the first times when I looked at data and thought, gosh, there is a problem here. We don't have enough data to talk about spatial variation. So what we saw was more variation in time and space, very strong negative autocorrelations that seem to be synchronized across populations. And we also saw this funny feature of the data whereby population growth rate declined through time. We thought that this was probably due to the fact that when demographers put in their plots, they tend to put in plots into populations that are doing really, really well, and there's only one way from there, and that's for population performance to decline. So that might be a bias in the way that we do demography. I've worked with Rob Salguero Gomez, who's led the curation of the Compadre plant matrix database, which has really done great work in putting together, collating and curating between species data on population performance. So we have good data now for nearly 1,000 plant species, and the equivalent for animal species as well in the Compadre database. But again, the same problems plague Compadre that we saw in our 2010 paper, and that's that there isn't much spatial variation between populations of the same species. So while we have really good between species information and can do really nice comparative demography, one problem is that we only have one or two or a few matrices, a few populations representing each species. And I'm really interested, in order to interpret this between species data, we need to know what's going on within a species to see whether the representative matrices that we're using in cross-species analyses, like the ones we do in Compadre, are actually appropriate, or whether there's a cloud of variation around that species point, which we're not taking into account. So the next sort of step in the journey, so we've done the 2010 paper, we've worked a lot using Compadre, we were using Compadre to try and work out what the environmental drivers are between species of population performance. This is some work from a postdoc, Sean Coutts, who's now at the University of Sheffield, asking whether we can use climate, geography, and phylogeny to predict population performance between species. And for this, we had a really nice sample size, 210 species this time. Over half of those had more than one population per species, and they came from a wide range of different families. But Sean put together this really nice graph, which just shows this problem in great detail, that if we look at the maximum distance between populations for where we have multiple populations per species, you can see that the vast majority of species just have populations within 1 or 2 kilometers of each other. So these aren't really good spatial replicates. We have a few brave souls out here who have looked at multiple populations and separated at a continental scale, but very, very few. And almost all of our knowledge of within species demography in Compadre comes from spatial replicates up to 20 kilometers. Now, this is just the data that's in Compadre as a nice way of sort of showing the global problem. I know of some excellent studies on garlic mustard, on the cushion plants, on Arabidopsis, which take a much broader spatial extent, but they just aren't included in the Compadre database for now. And what we see using Sean's work is that our capacity to predict various measures of population performance really decays quite fast. When you go beyond 20 kilometers away from a known population, you're no longer able to predict what an unknown population of an unknown species or even a population of the same species is actually going to do. So the data that we have currently are not really adequate for making predictions of what a population in a new place is how that population is going to perform. So the key phrase here in Sean's paper was, if you want to extrapolate demography with climate, proximity, and phylogeny, approach with caution. We're only able to do very limited extrapolation from our current between species data. Okay, so now just moving on, the next chapter of the story was we were trying to get more and more sophisticated in how we predict population performance. And this is work by Anna Chergo, who's sitting down there, around whether we can use probability of occurrence models, these are species distribution models built using climate data, whether we can use these to make predictions about population performance. And again, we were using data from Compadre, Anna built, with our collaborators in Switzerland, Antoine Guizan and Olivier Brunemann, built species distribution models. So, and these are populations in compadre. Oops. And here she built climate suitability surfaces. The green colors are areas that are highly climatically suitable for the species, where we predict a high probability of occurrence. And then these yellow to pink areas are less climatically suitable, where we predict a much lower probability of occurrence. We can situate some of our compadre populations in here in this climate suitability space and ask questions around whether population performance is correlated with climate suitability. So this is where we're building climate suitability models predicting probability of occurrence from occurrence data, and by using bioclimatic predictors, can we then take those predicted probabilities of occurrence and put them in as explanatory variables in order to predict population performance. So that just shows you that we're using compadre data. There we go. Compadre data for the demographic performance for 14 trees, 23 herbaceous perennials, and 37 species in total. The reason why we use 37 species is there's a limited suite of species for which we have location data and for which we can get really, really high quality climate-driven species distribution models. So we tried to get the best, highest quality data set we could for this. There's a whole bunch of reasons. Well, first of all, I guess the naive assumption would be that as climate suitability increases, population growth rate or some other measure of population performance should also increase. But there are a bunch of reasons. Ecology is the big reason why we expect populations to deviate from that expected relationship. So down here, we might expect, even though the climate is highly suitable, biotic interactions like pathogens or herbivores or competitors might actually be limiting population growth rate or population performance, and not the climate. And up here, even in a very climatically unsuitable area, we might get persistence of populations in favorable microhabitats, for example. So in fact, we do find every possible scenario when we look at the data. So here we have in mimulus, we have population growth rate is the size of these circles. This one seems to have read the textbook and is doing the right thing. So high population growth rate in highly climatically suitable areas, and low population growth rate out here in a low suitability climate. Primula here is doing everything in highly suitable climate, so it can have high or low population growth rate. And here with Saponaria, we find high population growth rate, so good population performance, even in climatically fairly marginal or unsuitable areas. So, and that's borne out by the data. We don't see a nice positive relationship between climate suitability and population growth rate, for example. However, we dug a bit deeper, and when you actually look at correlations between climate suitability and mean demographic performance in terms of fecundity, progression, stasis, and retrogression, or climate suitability and variability in demographic performance, we can actually find some relationships. And we then looked at how time to quality extinction is correlated with your mean demographic performance and the variability in demographic performance. And using this method, we found two pathways, basically. One is a resistance pathway, whereby plant populations can resist extinction in low suitability climates. So in low suitability climates, retrogression is higher, so plants can move back a stage and survive. And higher retrogression is associated with longer times to extinction. So you can delay extinction by having this retrogression strategy. And then there's a vulnerability pathway as well. So in low suitability climates, you're vulnerable to extinction through this pathway. And there's more variation in fecundity and progression in low suitability climates. And this is correlated with, higher variation in progression is correlated with shorter times to extinction. So you're more vulnerable to extinction through this pathway. So this was done using the 37 species that I mentioned. But it seems like this is basically setting up a hypothesis, two hypotheses that can be tested using within species variation in particular. We're not sure how strongly this holds for which species, which life history strategy. Okay. So if we can work out what enables populations to persist in these low suitability climates, that enables us to put management strategies in place, which can enable those populations to persist for longer, gives them more time to adapt to the new climatic conditions or the new environmental conditions, and gives us more time to maintain ecosystem services that may be vulnerable to climate change effects. And again, if we can work out what causes populations to be vulnerable to extinction in low suitability climates, this can throw up, again, management targets for maintaining populations in those low suitability climates through support in bad years, for example. Okay, so hopefully I've shown some of the reasons why I think it's really, really important to study within species variation in population performance and life history strategy. We really lack sampling over large spatial extents. We lack sampling over long time series as well. We found that space for time substitution is really inadequate given the data that we currently have, and this needs to be more thoroughly tested. We have confounding of space and climate, so two populations that are close together are likely to share environmental conditions, as well as being able to be connected via gene flow and dispersal processes. So it's not possible to tease those two things apart very easily. And we have difficulty distinguishing between the effects of multiple different drivers of population performance, the biotic and several different biotic and abiotic dimensions that control population performance. So this is where PLANTPOPNET comes in. With PLANTPOPNET, we're able to drastically expand this spatial extent out to 13,000 kilometers, which happens to be the distance between my PLANTPOPNET site in Kanturk and in Cork, here in Ireland, and another PLANTPOPNET site out in New Zealand, Bonnie Grubman, 13,000 kilometers apart, which is quite nice. Those two sites actually share a fairly similar environment. So while they're spatially really, really far apart, they're environmentally quite close together, which is really interesting. So we're starting to see the breaking apart of the confounding of space and environmental gradients. So this is the current state of PLANTPOPNET, our globally distributed demographic network. We have population genomics, which you'll hear more about today. Our field data, which we'll be talking about all week, which is the core of PLANTPOPNET, and the Common Garden experiment that Yvonne has done in collaboration with Jesus Vilelas, my postdoc. All of these orange dots are our field sites, and the red dots are the populations we currently have population genomic data from. So this is super exciting. We've grown massively. I'm going to give you a bit of the history. So the history started. Who remembers the beer in St. Lucy's? Yeah, PLANTPOPNET started over a beer in St. Lucy's Cafe in Brisbane, Australia. I think it was 2012, but I might be wrong. It might be 2011, 2012. When a bunch of us got together, we'd been working for some time as part of a working group, a population dynamics working group. And we just kind of proposed one day. I thought, well, if I had got some experience with distributed networks through NutNet, the nutrient network, and thought we really needed this for plant population dynamics. So I just plied them with beer and got them to agree. The first steering meeting, when it really kicked off, was in 2014. We had a meeting here in Trinity College Dublin, where we got the steering committee together, and we discussed the protocol, and we put in place some of the governance around PLANTPOPNET, and we all undertook to start some sites, and also to persuade some of our friends to start sites. So that's how it started out. And the first field data that we have is actually from Elizabeth's site, and that started in 2014. And this is where we are now. So just 3 years later, it's grown hugely, beyond my wildest dreams. I have pretty wild dreams. This is beyond them. We have 68 sites with at least 1 year of data, so 48 native sites and 20 non-native sites, which is just fantastic. My wildest dreams, I thought, if we can get 30 sites, that would be great, because 30 seems to be a magic number in ecology. If we got 50 sites, that would be awesome. 68 sites is just fantastic. We have 35 sites with at least 2 years of data. So these 35 sites are the ones you can play with now to build some models this week if you want. Again, 24 native, 11 non-native, so a really nice spread. Oh, sorry, that's not 35 now. What is it, Alan? That's 43 as of this morning. 43 as of this morning. we had some late data entry last night from some of our participants. So thanks to them for pulling out all the stops to get the extra data into it. So that's fantastic. We have 11 sites with at least 3 years of data. So now we can start to build stochastic population models. And we have one site with 4 years. So hopefully, what I'm hoping for is that we get progression of all these sites into the 4 and even 5 year category, so that we end up with at least 50 sites with 5 years of data, which will represent the most amazing, spatially extensive demographic data set that I could ever have imagined, and will just give us toys to play with for the next decade, which is great. And 16 countries, so we're doing pretty well. If anyone knows anyone in South America or Africa who fancies being part of this network, we'd love to hear from them, or Asia. So I totted up using my estimate of around 4 person days per year for getting the core demographic field data together, multiplied by the number of sites, and came up with an approximate 520 person days of field demographic effort. So this represents about 2.5 years of one person working full-time just in the field collecting data. So that's a pretty amazing amount of field effort that's being contributed by our participants. My next job is to try and put a dollar or a euro sign against that, and this is a really great resource, one that would be incredibly hard to collect again in any other way. This is the value of the distributed approach. That does not include the hours that we spend in coordination, the hours that Alan in particular spends in data curation, the analysis that the postdocs and PhD students are doing, and the writing of papers. So we have a couple of draft papers now that are we're ready to talk about, and there's been some effort gone into writing of those already. So that's really exciting, and it is a big project. It's something pretty major. How do we fund all this? I started out with a very small amount of funding through a Marie Curie Actions Career Integration Grant when I moved to Dublin. So I think approximately around 15,000 of that went to some of Alan's time in order to coordinate sites and curate data. I was lucky enough to get a Science Foundation Ireland grant in 2015, and I reckon around 200,000 of that has been spent on postdoc time, Alan's time to curate data, the genomic analysis, and the Common Garden experiment. So it's funded some of the extra bits and pieces around the network. And then, of course, all of you who, like I said, have contributed so much time and effort, and who've gone out and found funding yourselves for your sites, or just begged, borrowed, or stole time and resources from wherever you could in order to get the data to us. So that's how it's been funded. We're PLANTPOPNET HQ. So that's myself as coordinator, Alan Finn as my research assistant, who deals with all the data curation and coordination, deals with the field protocol and organizes all of our field work here in Ireland and overseas. Annabelle Smith, who does the population genomics. Jesus Vilelas, who does trait variation and population modeling, and you'll hear more from both of them today. Maud Bodraz, who's just started a PhD and has already completed one season of fieldwork in the Swiss Alps. Plantago, she is coordinating, which you'll hear more about, I think, tomorrow. And Anna Chergo, who has been on the steering committee since the start and is now funded as a postdoc, and she's helping out with the species distribution modeling and general work around trait variation. So that's us, and this is you. So these are some of our participants, some of our site coordinators. Oh, I think it was blanked out. That was a picture of me. I didn't remove it on purpose.
Stand there with your head right.
That's me. So these are just some of the pictures that you sent through to Alan and Annabelle, which we put in a montage here. So this is all of you. 43 site coordinators, which is just fantastic. And our steering committee, of course, which came together first in 2014. And the steering committee will be meeting throughout this week as well to progress the direction of PLANTPOPNET, the protocol and the governance issues around it. So that's who we all are. Now, onto the aims for the workshop. The first thing that we really wanted to do and use this opportunity to do in person and remotely is really to update all of the participants in the steering committee and everyone associated with PLANTPOPNET on our progress to date. I think we've made a lot of progress, but we haven't sort of packaged it all up and put it together in one place until this week. So this has been a fantastic opportunity for all of us to basically do that and produce these resources. We want to get your contributions, both the people here who've come in person and the people who are participating remotely. We need your contributions to make these papers better and to get them up to a really high standard. We want to progress new analyses, old analyses, as well as new analyses, ongoing analyses, and models. So getting ideas and help in progressing the science of what we want to do. We will also have some sessions on reviewing the protocol, making sure we're all doing the same thing in all the sites, troubleshooting issues. There are a few issues that we need to talk about. We need to talk about clones, we need to talk about seed banks, the tricky things that we kind of conveniently ignored up until now. We're going to have steering group meetings, as I mentioned. I'm hoping that we'll really use this opportunity to get new ideas and opportunities for the network. And we want to strengthen the network, provide you all with the resources you need to write grants, to get people working on the data. The data belongs to all of us, and I don't want all of the scientific progress to be led from the headquarters. If you have a great idea for an abstract for a paper that uses the PLANTPOPNET data, and now is the time to put your hand up and say, I would really like to progress this. We can talk about it while we're here and partition out some of the work. That would be fantastic. And just want to wrap up by saying many, many thanks to all of our participants. We couldn't have gotten this far without all of you. So thanks very much.